Now this last video is on the biological molecule, which is chapter 2, but it is the third video on this chapter, and it's the syllabus 9700, and we're going to discuss syllabus points 2.3 and subdivisions of it 4, 5, 6, 7 and 8, and 2.4, which is about water. The first thing to understand is globular proteins. Now, globular proteins are generally, the most important thing is that globular proteins are soluble. And how are they soluble is, number one, they're globular. Doesn't mean that we say the word globe-like, because that's in the question. So you can say they're round, you can say they're spherical, you can say they're ball-shaped. So any other word but the word globe-like. So globular proteins are round, and an example of that is hemoglobin, and all the enzymes that we have in all living organisms, whether they are plants or animals. Now the fact that they are soluble is a very important reason, is because if they are round, the protein has been folded in such a way that all the hydrophilic R groups are on the outside and the hydrophobic R groups are on the inside. You know, naturally, if it's going to be transported in the plasma, or it is present in the cytoplasm of the cell, any cell, whether it's a plant cell or an animal cell, every cell cytoplasm is 70% water, roughly 70%, some could be more or less. So the fact that all these globular proteins have a physiological roles means they are important in the functioning of the living organism. So in order for them to function, we need enzymes. Any living organism needs enzymes. Even if it's a bacteria, it needs enzymes. Even if it's a human, it needs enzymes. Even if it's a mango tree, it needs enzymes. So globular proteins are round, globular proteins are soluble, and number 3, their hydrophilic R groups are on the outside and the hydrophobic R groups are on the inside. And the one that we have to study is hemoglobin. Now hemoglobin has a quaternary structure. Why does it have a quaternary structure? Because it has 4 polypeptide chains, and there are 2 types. They are not 4 different ones. Like if I had said there were 4 different ones and one was alpha, beta, gamma or theta or something like this. No, but I am saying 2 alpha, 2 beta. So there are only 2 types. So besides this, you see, every protein is made of amino acids. So it means the primary structure of the alpha chains is the same, and the beta chains, the primary structure of the beta chains is the same. So this is the beta chains, and this is the beta chain, and this is the beta chain. So the primary structure means the sequence and number of amino acids would be the same. So this one would be the same and this one would be the same. Similarly, the alpha ones would be the same, but the alpha and beta would be different. Then besides the protein part, we call this is called, there is a prosthetic part. Prosthetic means it's not its own. Prosthesis, dental prosthesis, people who wear artificial teeth. So there is an iron part, and that iron part, which contains Fe2+, is called the heme part, and that's the word which gives it hemoglobin. So the 4 polypeptide chains, so quaternary structure, hemoglobin has a non-protein part which is called the heme part and which contains iron. Now this is another diagram in which you can see there is a red blood cell, and from the red blood cell we have extracted the hemoglobin. And the fact that you can see how this diagram shows you how that protein's folding has taken place, and you see a lot of these alpha helixes. And then you can see the iron part, which is the green part, and the hemoglobin combines with oxygen to form oxyhemoglobin, and that iron combines with 4 oxygen molecules or 8 oxygen atoms, and that is how, now this is a reversible reaction. In the lungs, oxyhemoglobin is formed. In the body cells, the oxygen is given up. So in the body cells, the oxygen will be given up. In the lungs, the oxyhemoglobin will be formed. That's why we say it's an unstable compound. And then in the body cells, the oxyhemoglobin gives up its oxygen so that the cells can use it for aerobic respiration. Next protein, or the group of protein that we have to study, is called a fibrous protein. So one that we studied was the globular protein, in which we said they are round, in which we said they are enzymes, and there's hemoglobin, and they are antibodies, myoglobin. But besides this globular protein, there's another type of protein which is called a fibrous protein. And the fibrous protein is insoluble because it has a different role to play. It has a role of a structural role, and you can see where collagen is found. It's found in hair, it's found in connective tissue, it's found in the skin, and it is found in cartilage, which is at the ends of the bones. Then it's found inside bone, then it's found in your nails. So fibrous protein has a different role to play. It is actually going to be needed to strengthen certain parts of the body where we need to strengthen, like the bones have to be very, very strong because they have to take all the pressure, the weight, joints. When you stand up, all the weight goes onto your weight bearing joints, which is your knee joints. So what you have to understand is that fibrous proteins have a different role to play. They are not like globular proteins. So fibrous proteins example which we have to study is collagen. Now we have to know a little more detail about collagen. What is it made up of? Now the first thing that we have to understand about collagen is that it is a triple helix. So it is made up of 3 polypeptide chains, and these 3 polypeptide chains are twisted around each other. And every third amino acid is glycine. And if you remember, when I was doing the different amino acids with you, I told you to remember this because its R group is H, just hydrogen, nothing else. And it's the smallest amino acid. So the reason that it is the smallest is helps us to make it easier for the 3 polypeptide chains to twist around each other. Now you have to understand is that this is basically going to give strength to the bones. So it's got to be something which is going to be very, very strong. So the collagen molecule is made up of 3 polypeptide chains, and then it's got, in between the chains, it's got hydrogen bonds and covalent bonds. So there are covalent bonds present which are joining these polypeptide chains to each other. And then there is another very important phenomena which is called staggered ends. Staggered ends. Now you see, this one ends here, then it ends a little lower, then it ends a little lower. So you have this weak area like this. So this is called staggered ends. If you look at a brick wall, you see how the bricks are placed. Now the bricks are placed, they're not placed one upon each other, because if you had the bricks like this, like for instance if you had them like this, and then again you had them like this, now you see there would be this weak area here. Now when you look at bricks, they're never placed like this. They're always placed, one ends here and then the other one ends here. So similarly the triple helix, now I've shown you one of these, and this ends here, and then the other one starts here. But in the other row, this one ends here, and then this one ends here. Now the fact that they don't end at one place, this is called staggered ends. And this is very important because this gives it immense tensile strength. So collagen is present in tendons, is present in cartilage, is present in the wall of the arteries, which has to withstand a lot of pressure. So all this helps in making them very, very strong. We must be very clear on two terminologies. One is called the collagen molecule, and the other is called collagen fibers. And you can see here this whole structure, the triple helix. And please remember, this is not the alpha helix which we study in the secondary structure of proteins. This is called a collagen molecule, and then all of these collagen molecules pack together and form microfibrils. So the difference between collagen molecule and the collagen fibers should be very clear to everybody. And their function, of course, is giving it the structural role which I talked about. Now, why is it that collagen is one of the biggest topics which is under research these days? Is because what happens to the skin as you age, it starts to wrinkle. And I'm sure none of us likes it when we see those wrinkles on our face. So you can see the difference between a young skin and an elderly skin. So why are we doing so much research these days on collagen is because if we can prevent the loss of collagen results in wrinkling. So if we can develop some sort of a medicine or we can prevent it, so we will be able to prevent wrinkling, and that is what goes on into all the anti-wrinkling creams which people use, and that prevents the wrinkles from becoming worse as one ages. So a lot of research goes on into why collagen is lost as a person ages, but we've not actually been able to figure it out. The next, this is please pause the video here and then look at this and make your notes. I've given you two comparative tables between fibrous and globular proteins, and they're very, very interesting. One is compact, extended, soluble, insoluble. Secondary structure is complex with a mixture of alpha and beta. Secondary structure is simple, based on one type only. Quaternary structure is held together by non-covalent forces. Here it is held together by mainly covalent bonds. And functions, of course, enzymes, transport, immune protection like antibodies and hormones. So we have to understand then fibrous protein, as we've talked about, tendons, ligaments, hair and skin. So this is another table which gives it in a little different manner. A class of insoluble proteins. Soluble has a long narrow shape, has a round spherical shape, have a helical or sheet structure, have a folded ball-like structure. In the secondary structure, in the tertiary structure, consist of repetitive amino acid sequence. You see, fibrous proteins are just, so they have a repetitive structure. But these consist of an irregular. Why? Because if you remember, hemoglobin has 2 alpha and 2 beta chains. So it's not a very repetitive structure. It could be a little different, especially when we have it in enzymes. Then insoluble, soluble, then have strong intermolecular interactions, weaker ones, less sensitive to changes in temperature and pH. Why? Because they're mainly acting as a strengthening molecule. But these, of course, are very, very sensitive, like enzymes are very sensitive to changes in temperature and pH. And these are involved in forming structures like bones and ligaments. These are all involved in metabolic reactions. Metabolic means that they are important in, like enzymes are there to lower the activation energy in every reaction. So without it, the cells just won't function. And then, of course, you have the examples. Now we come to the same chapter, which is biological molecules. This 2.4 is about water. Now we have to know the structure of water and why is it a very good solvent. So it is the ability to dissolve. Water is a universal solvent, and we have to understand why is its structure so important. Now individual water molecule is bent, and this angle is about 105 degrees, and that gives it charge. So we say water is a dipole. So there is a partial positive charge and there is a partial negative charge. Now if you look at the water molecule, there's hydrogen bonds formed between water molecules, and this of course is very important because water then moves as a fluid, and we will be talking about cohesion and adhesion in later chapters. So the fact that the water molecule is polar and there is a partial negative charge on the oxygen and partial positive charge on the hydrogen, that of course is a very helpful thing because it helps to form hydrogen bonds between water molecules. So that's why the water moves as a continuous liquid. But then you see how it forms. How is it a solvent? It's a solvent for ions. It is a solvent for polar molecules. So you can see how sodium is very quick to dissolve in water. So sodium and chloride ions. So sodium ions, chloride ions, all dissolve very rapidly in water, and the reason for that is because you see how the water is orienting around it. The oxygen is facing inwards because sodium is positively charged. Now it will be the other way around for chloride ions. So please draw a diagram to show how chloride ions would orient and how the water would orient around it. That's for you to figure it out. And the second property of water that we need to discuss is high specific heat capacity. High specific heat capacity. Now that's a very important for in biology. Why? Because every human being is 70 to 80% water. Plants are water, bacteria is made up of water, fungus is made up of water. Now what is the definition of specific heat? As it says here, the amount of energy it takes to raise the temperature of a substance by 1 degree Celsius. And we say water has a very high specific heat. Now that is very important. I mean, look at the situation in which we would have said water has a low specific heat. Now that would have been bad. So this is important is because now what will happen is the ocean does not drastically change temperature. So, and what does it mean high specific heat? This means that compared to other substances, it takes a big change in energy to change the temperature of water. And specific heat examples, as we can see here, is a lake's large mass and water's high specific heat prevents the lake's temperature from changing quickly. So it doesn't get hot very quickly, it doesn't get cold very quickly. But you look at other things like iron and aluminium have low specific heats because they heat up quickly, they make good cooking utensils. And of course we have this lovely fish who's happy because she can survive or he can survive is because the water has a high specific heat capacity. So fish can survive in water. Even other aquatic organisms can survive in water because water would not heat up. Even if the sun is shining very brightly, water is not going to heat up. Now the last topic is high latent heat of vaporization. The property of water, the fact that it is high latent heat of vaporization is a very, now please remember, it's the word high has to be understood. Now, as water evaporates, the surface of the water that remains behind cools. Now, what happens? What is latent heat of vaporization? Now, you see here, energy must be supplied to break the hydrogen bonds between water molecules. So, you come into this room is very hot, and immediately your skin senses it, and all this impulse goes to the brain, and the brain sends impulses for the sweat glands to start producing sweat. Now, you have to understand production of sweat is not going to cool you down. The sweat must evaporate. And the principle behind sweating is to maintain the body temperature of land animals. So our body temperature, I enter this room is very hot, but my body temperature is going to remain at 37. So the fact that when you enter some place and it's hot and sweat production occurs, and then the sweat evaporates. When it evaporates, it actually takes the heat from your body. So you might come into the room and you say, oh, bahut garmi hai. And then you switch on the fan and you switch on the AC. And of course, all that is going to help in the evaporation of sweat. Just a point that I want to make about is that why does ice float? Now, usually, you take some water and you just take some ice out of the fridge and just put it in the water. Well, it's going to float. Why does ice float? And the reason we say is it is less dense. Now, you see how the water molecules have sort of arranged themselves, and they've arranged themselves, spacing them. Now, this is water, liquid, that's steam, and that's ice. So this wording, this terminology that we use is that ice floats because it's less dense. And of course, that has a very important biological impact. You see, when ice floats, then the water underneath is going to remain liquid, because all this ice which is formed is going to come to the surface, and it sort of forms an insulating layer. And in the water deeper down, the fish can survive and they won't die. So this is just a point of a biological point which I have explained to you, is why do we say these things? Why do we say less dense? So this is just a little bit of biological English, though it's not in the syllabus anymore that we talk about the fact that ice becomes less dense and it floats. It was in a previous syllabus. Okay, that is the end of this chapter. Please revise this chapter and then let me know what your problems are. Thank you.